This is the lecture for chapter 14 for microbiology. We're looking at non-specific host defenses. Let's do a chapter overview. And so scoping out chapter. So an obstacle course for microbes. So much of the body is protected by barriers that stop or deflect microbes near the body surfaces before they get too far. One example of these defenses is cilia in the trachea that brush foreign matter out of the respiratory system. We'll talk more about that. So host defenses. So first off, you have innate, which are non-specific, and then you have acquired, which are specific. We'll get into that with chapter 15. But for innate or non-specific, the first line of defense is physical barriers, chemical barriers, and then genetic barriers. And then the second line of defense is inflammatory response, interferons, phagocytosis, and complement. Then that's when you get into acquired. That's the third line of defense. That's the B T lymphocytes, the antibodies, cytotoxicity. So some defenses are non-specific and present at birth, and others are specific and must be acquired over time. But all are essential for complete immune protection. In this chapter, we will survey the anatomy and physiology of the inborn and non-specific defenses that set up the immediate zones of protection. We'll talk about neutrophils, which are phagocytes. We'll talk about basophils. They function in inflammatory and allergies. And eosinophils. They're active in worm and fungal infections, also in inflammation and allergies as well. And we're going to look at the origins of immunity. So looking at blood cells, you have red blood cells and white blood cells. So tissues in the bone marrow produce several different types of white blood cells that become the basis of our defense system. Some of them are phagocytes. Some function during inflammation. Some react against specific infectious agents. So we're going to talk about these wandering search parties. So most blood cells are highly active and modal with an ability to seek out and recognize abnormal or harmful cells and substances. There are also guides that recruit other cells to fight off infections. We also have a flood of protection. So when inflammation strikes, an injury to tissues and cells triggers a spontaneous series of reactions at the site of the damage. This event will alter the size and permeability. That's how easily things go across nearby blood vessels, stimulate migration of white blood cells to that area, and cause an outpouring of chemical substances. And those are all designed to contain pathogens and to stimulate immunities. Then we're going to talk about phagocytes. These are powerful predators, and it's a special breed of white blood cells stationed throughout the body to actively pursue foreign intruders, swallow them up, and kill and dismantle them. This here is a phagocyte, and these are like microorganisms, and the phagocyte will take it in and it will destroy it. So, overview of the host defense mechanisms. First, you have innate. This is the natural defenses. These are defenses that are present at birth, and they provide non-specific resistance to infection. That means that they're not specifically targeted at one thing. They will protect against anything that's not normal for your body. Then we have adaptive immunities. These are specific, and these must be acquired. So here's a diagram that talks about the line of defense. With the first line of defense, that's innate, which means you have it from birth. It's non-specific, and you don't have to, it does not develop a memory. So here's some examples: skin, tears, coughing, sneezing, chemical barriers like low pH, lysozyme, digestive enzymes. Then you have genetic barriers as well, resistance that are inherent in genetic makeup of the host. So somehow the pathogen can't get beyond the genetic makeup. It's almost like someone that is sickle cell anemia. If they have the trait for sickle cell anemia, that trait, and they're not going to catch malaria. That's the first line of defense. Second line of defense is innate as well. It's not acquired. It's mostly non-specific, and so it does not develop a memory. These are things like phagocytosis. There's certain cells that are just in our body, white blood cells, that will engulf foreign particles, things that shouldn't be there. Inflammation, fever, interferon, complement. The third line of defense is acquired. This is specific, and so it's specific for a certain microorganism or a certain invader. You can develop a memory of that. And this is where you have the T lymphocytes, B lymphocytes, and the antibodies. So defense mechanisms of the host. To protect the body against pathogens, the immune system relies on a multi-level network of physical barriers, immunologically active cells, and a variety of chemicals. So the first line of defense, any barrier that blocks invasion at the portal of entry, that's non-specific. So your skin it blocks entry of viruses, bacteria, any contaminant. As long as it's intact skin, that is a non-specific first line of defense. Second line of defense, these are protective cells and protective fluids like inflammation and phagocytosis. They're non-specific. Once again, they aren't targeting a specific intruder. They just will stop whoever should not be there. Then you have the third line of defense. This is acquired with exposure to a foreign substance. Now, this is where you provide protective antibodies and creates memory cells. This is very specific. When you actually come across a foreign invader, the body builds up immunity to it, and then your body remembers that specific microorganism or invader. So the lines of defense do not work separately. So most overlap and are redundant in some of their effects. It means they overlap. First line of defense, the barriers at the port of entry. So a number of defenses are a normal part of the body's anatomy and physiology. So these are inborn or innate. They're non-specific defenses. They can be divided into three categories: physical or anatomical barriers at the body surface. They could be chemical defenses. They could be genetic resistance to infection, and they could block they block the entry of not only microbes but any foreign agent, whether it's living or not. So here's some of the built-in defenses of our skin. So the outermost layer of the skin has epithelial cells. Oops. They're cemented together and impregnated with keratin. Then another built-in defense of the skin is you have a flushing effect of sweat. Then you have mucous membranes. So the coating of digestive, genital urinary, and respiratory tracts are coated with mucous membranes. Then you have blinking and tear production. Then you have flow of saliva. Then you have flushing effect of urination, defecation, and vomiting. Those are all physical or anatomical barriers. Okay. And this just shows, has a diagram that describes some of them. Like the tears have lysozymes in them. They will break down bacteria. You have sweat glands and sebaceous glands. They have fatty acids. You have mucus in the respiratory tract and the digestive tract. You have saliva. And you have your skin itself is a physical barrier. Then you have the normal microbiota. That's normal bacteria, viruses that are on your body that help protect you. You have low pH on our body, mucus once again in the respiratory system, then normal microbiota in the large intestine. You have different cells that are used for the immune system. Defecation, you physically pass bacteria and things out. Urination flushes your urethra as well. So mucous membranes, mucus coat impedes attachment and entry of bacteria. They get stuck into the mucus and they don't make their way all the way in. Then you have ciliary defense of respiratory tree. So first off, you have nasal hair that traps larger particles in your nose. That's nasal hair. As you get down further into the trachea, down in the respiratory tract, you have cilia. Cilia move in an undulating fashion. They keep flowing and pushing things up. And you have what you call the mucociliary escalator. It keeps pushing mucus up, not getting into your lungs, and then you cough up, spit it out, or swallow it. That's the mucociliary escalator. So this just describes it a little bit closer. It shows nasal cavity has mucus on it, nostril oral cavity, there's mucus, the pharynx, epiglottis, you have a lot of mucus in all of these different areas. So some of the non-specific chemical defenses. So from the skin and mucous membranes, you have sebaceous secretions, and then you have antimicrobial secretions from specialized glands. That's in your skin and mucous membranes. You have antimicrobial secretions from specialized glands and also sebaceous secretions. Then you have other defenses in tears, saliva, and skin. We have what we call lysozyme. That's an enzyme that hydrolyzes the cell wall of bacteria. Hydrolyzes is breaking down the cell wall using water. So it breaks apart the cell wall. And you have defensins, which are peptides that lyse bacteria and fungi. Then you have high lactic acid and electrolyte concentration in sweat. So lactic acid, which means the pH is low, and that's the skin's acidic pH, is a chemical defense. Then you have hydrochloric acid in the stomach, very high, at very low pH, high acid level in the stomach. Then you have digestive juices and bile of the intestines. That's another chemical defense. And then also semen contains an antimicrobial chemical. And then the vagina has an acidic pH, so it's very low pH. That's maintained by the microorganisms in the vagina. And then we have genetic defenses. Genetic defenses means that some hosts are genetically immune to diseases of other hosts. So an example of this is humans can't acquire distemper from cats, and cats can't get mumps from humans. So that's saying that there's certain microorganisms that, because we don't have the same genetic makeup as a cat, we can't catch all of the diseases that a cat can catch. So viruses have great specificity for their host receptors. Viruses are very specific as to what type of organism that they can infect. So some genetic differences and susceptibility exist for other pathogens, including differences within members of the same species. That's where I was talking about the difference with the sickle cell anemia. For some humans carrying a gene or genes for sickle cell anemia, they're resistant to malaria. So that's a genetic defense. So, which of the following is not an example of the immune system first line of defense? You include that inflammation, lysozyme, hydrochloric acid, stomach, sneeze reflex, cilia in the respiratory tract. All these are, except for inflammation. It's not the first line. So structure and function of the organs of defense and immunity. So immunology is the study of the body's second and third lines of defense. So the primary functions of a healthy immune system. First off, you have surveillance of the body, recognition and differentiation of normal versus foreign material, and then attack against and destruction of entities deemed to be foreign. So primary functions of the healthy immune system, surveillance of the body. That means you have a lot of patrols that are going around in your body all the time. That's healthy immune system has substances that go through your body, go through your tissue, and they're monitoring your fluids and tissues all the time, looking for anything that's foreign. Then you can recognize and differentiate between normal versus foreign material. So they recognize and they say, wait a minute, this is normal, this is not normal. And then they are able to attack against the destruction of entities deemed to be foreign. So, white blood cells, which are called leukocytes, they have innate capacity to recognize, which means an inbred and inborn capacity to recognize and differentiate foreign material. So anything that is non-self is considered foreign. Anything that's self are normal cells of the body. And I'm not going to ask you about pathogen-associated patterns and pathogen recognition receptors. So all this is mainly carried out by our white blood cells, which are the leukocytes. They have the innate capacity to recognize and differentiate any foreign material. So they know that something that is non-self is foreign. Self is normal cells of the body. And once again, they talk about pathogen-associated patterns and pathogen recognition patterns, receptors. What it is certain pathogens. Let's see. So here's a molecule that has self antigens on it. This is a white blood cell, and it can recognize that that's self, so it won't attack it. This has this white blood cell recognizes that does not have, it's not self, so it has antigens on it. Once it recognizes that it has antigens, so it's not self, then it engulfs it and eats it up, destroys it. So the components and connections of the immune system. The immune system is a large, complex, and diffuse network of cells and fluids that penetrate into every organ and tissue. So the body compartments that participate in immune function, reticular endothelial system. This is between the blood and the cells. And so all the tissue that's extracellular, that's outside of the blood vessels, outside of the cells, but it's everything in between. Then you have extracellular fluid, then you have the actual bloodstream, and you have the lymphatic system. So for effective immune responsiveness, the activities in one fluid compartment must be conveyed to other compartments. This is where it shows this is a blood vessel, a capillary, and this is a lymphatic vessel. These are cells. Whatever tissue this is, these are tissue cells. This is the extracellular space. And so this is reticular endothelial um, area. It's outside of the blood vessel, it's outside the cell, and it's everything in between here. So what you have is you have white blood cells that are able to pass across the capillary, get into this space, and actually go into the lymphatic area. And they're just constantly searching out and seeing if there's any abnormal cells present. So reticular endothelial system is a network of connective tissue fibers that interconnects other cells and meshes with the connective tissue network surrounding organs. So this area is inhabited by phagocytic cells, which are cells that can engulf foreign things or inoperable tissue cells. And macrophages are ready to attack and ingest microbes that pass the first line of defense. And so this is the same. This is cells. This is the reticular endothelial are reticular cells. They're outside of the blood vessel, but outside the cell as well. This is a white blood cell, and these are white blood cells. And then uh, when you have something that is abnormal in here, they're constantly looking around and searching around to actually kill something that is not should not be there. Now, the origin, composition, and functions of blood. So, whole blood is plasma and the formed elements, which are the blood cells. Then you have serum, is the liquid portion of blood after a clot has formed. So it has minus clotting factors. And then you have plasma. Plasma is 92% water, has metabolic proteins, globulins, clotting factors, hormones, and other chemicals and gases to support normal physiological functions. This is where we talk about hematocrit. You have the red blood cells that make up the largest portion of the formed elements. You have the buffy coat, which makes up the white blood cells and the platelets. And then you have the plasma, which is mostly, vast majority of it's water. But in there you have, um, other proteins and hormones and all. That's unclotted blood. When blood clots up, then what's left up here is serum. And so this is the blood clot, and then the serum is what's left on the top. So now, hemopoiesis, also called hematopoiesis, is production of blood cells. So first you have stem cells, which are undifferentiated. So they're precursors to new blood cells. Now there's different types of blood cells that can be developed from these undifferentiated cells. You have primary cell lines are platelets, which are thrombocytes. You have red blood cells, RBCs. Leukocytes are white blood cells. It's the WBCs. The leukocytes or the white blood cells are what's responsible for immune function. They come from the same precursors that red blood cells come from, same precursors that platelets come from. You have two different types of white blood cells, granulocytes, which have lobed nucleus, and agranulocytes, which are unlobed, so the nucleus is rounded. This looks complicated, but it's not as bad as it looks. This is what starts with an undifferentiated precursor. And then this will divide out, and you see it will go here and will become red. Part of one cell line becomes red blood cells, so some of them are red blood cells. Another cell line goes and becomes platelets that's involved in blood clotting. Then another line will become the neutrophils, basophils, eosinophils. These are all the granulocytes. And this cell line will go and it will be an agranulocyte, but it will be monocyte specifically. Now, the same precursor that did all that, the same precursor could actually go a different direction, and it goes to the lymphoid stem cell. And these lymphoid stem cells will form the lymphoblasts, which become the lymphocytes. The lymphocytes are the ones that we will talk about in chapter 15, which um, has the adaptive or specific immunity. These are the ones that use the specific, these are the antibodies. All of these are non-specific immunity. These are non-specific. These will attack anything that's not like you, anything that's foreign. These are very specific for specific microorganisms. Okay, we'll talk about that later. They all come from the same precursor. So the neutrophils, they make up between 55 and 90% of the granulocytes, and they're lobed nuclei. And then eosinophils make up about 3%, 1 to 3%. The basophils is just 0.5%. These are some of the pictures of them. So that then you have those were the granulocytes. The agranulocytes, 20 to 35% are made up by the lymphocytes. Lymphocytes are the ones that are specific immune response. You have B lymphocytes and T lymphocytes. Then you have the monocytes or the macrophages. These are the ones that uh, make up the phagocytes. So they're the largest white blood cells. They're phagocytic. So you have macrophages and dendritic cells. Okay, here they kind of describe it a little bit more. Talk about the neutrophils. Uh, they're 55 to 90% of all the white blood cells. They're multi-lobed and they have purple granules that contain digestive enzymes. And they're primarily used for phagocytosis. Eosinophils, their primary function is destruction of parasitic worms, and they actually help with allergic response. And then you have basophils. They're very active in allergic and inflammation, and also with parasitic infections. The monocytes are used for phagocytosis. And so they differentiate into macrophages and dendritic cells. Then you have the lymphocytes, which make up 20 to 35% of the white blood cells. This is the specific or acquired immunity. And then two types of lymphocytes exist: T cells, so that's T lymphocytes, and B cells, B lymphocytes. So the erythrocyte is a red blood cell and developed from bone marrow stem cells, lose the nucleus, and then they contain sacs of hemoglobin. Then you have platelets, which are formed elements in circulating blood that are not whole cells. So you have erythrocytes, platelets, and then leukocytes, which are the white blood cells. Now you have the lymphatic system. This provides an auxiliary route for return of extracellular fluid to the circulatory system. This, when you remember in A&P, when it talks about the lymphatic system, brings any fluid that was not returned to the circulatory system, brings it back to the circulatory system. Acts as a drain-off system for inflammatory response, and it is useful in surveillance, recognition, and protection against foreign material. In the lymphatic system, it houses a lot of white blood cells. And it just talks about the lymphatic system, runs all through the body, and it sequesters fluid and any microorganisms that are in the body, and then brings it back into the circulatory system. So lymph is a plasma-like liquid carried by the lymphatic circulation. So like the plasma in the whole blood, the plasma portion of the whole blood is a lot like lymph. And so formed when blood components move out of blood vessels into extracellular space. It's made up of water, dissolved salts, and 2 to 5% protein. Transports white blood cells, fats, cellular debris, and infectious agents. You have lymphatic capillaries that go through all parts of the body except for the CNS, bone, and placenta. They have very, very permeable, so they're thin walls, very permeable by extracellular fluid, which is moved through contraction of skeletal muscles. So it returns the lymph, returns fluid to the regular circulation. It's a one direction flow, flows in one direction toward the heart, and eventually returns fluid to the bloodstream. And so this is like with the heart, sends blood out through the aorta, then goes through the arteries, arterioles, through the capillary bed, then blood comes back up through the veins, comes back to the heart. Well, some of that blood that leaves on the arterial side, some of that fluid does not go back in the venous side. That's where the lymphatic system picks up the fluid that was left in the tissue and it goes through lymph nodes. These are lymph nodes that is going up through, and these lymph nodes house white blood cells. So when fluid flows through there, the lymph nodes will identify something that's foreign and will mount a defensive attack. And then that fluid is returned back into circulation. So the finest level of lymphatic circulation begins with blind capillaries that pick up foreign matter from surrounding tissues, transport it in lymph away from the extremities via a system of small ducts. Ducts carry lymph into a circuit of larger ducts that ultimately flow into clusters of filtering organs, which are called the lymph nodes. Don't have to worry about that. This just shows that as the lymphatic vessels sequester fluid, then it goes through an area which is a lymph node. This is how a lymph node looks. This cross-section of lymph node, and it just houses a lot of white blood cells. So any fluid that comes in, they're able to identify anything that shouldn't be there. So classification of lymphoid organs. The primary organs, sites of lymphatic origin and maturation. So you have the thymus gland and the bone marrow. Those are primary organs in the lymphatic system. Secondary organs, circulatory-based locations like lymph nodes and the spleen. These are mucosal-associated lymphoid tissue, skin-associated lymphoid tissue, and gut-associated lymphoid tissue. Lymphoid organs, the thymus, high growth and activity until puberty. Then it begins to shrink. This is where T cells mature. So the thymus is where T cells mature. And it has high growth and activity until puberty. Then it begins to shrink. Then you have lymph nodes, which are bean-shaped organs, long lymphatic channels. Then you have the spleen. The spleen is structurally similar to lymph node and filters circulating blood to remove worn out red blood cells, and it removes pathogens. And it has a lot of white blood cells in it as well. So it literally filters out pathogens. It just shows the placement of the thymus gland. Okay, where do the T cells mature? Thymus. Where do B cells mature? Bone marrow. Okay, now let's look at that was all still part of the first line of defense. Let's go to the second line of defense. Inflammation. So mechanisms that play important roles in host defenses, you have to recognize that something's not right. Then you have inflammation. Then you have phagocytosis, which it engulfs it or eats it up. You have interferon and complement. So they're non-specific in their effects, but they support and interact with specific immune responses. So reaction to any traumatic event in tissues that attempts to restore homeostasis. That's what the inflammatory response is. The reaction to any traumatic event in the tissues that attempts to restore homeostasis. So inflammatory response helps to clear away invading microbes and cellular debris left by immune reactions. These are the four characteristic signs or symptoms of inflammation. You have redness, which is called rubor, just increased circulation and vasodilation in response to chemical mediators. So you get redness because you have a lot more blood flowing. Then you have warmth, which is also called calor. This is in the Latin terms. So heat is given off by the increased blood flow. Then you have swelling, which is called tumor in Latin. That's increased fluid in the tissue as blood vessels dilate. You get edema. Then you get white blood cells, microbes, debris, and fluid collect to form pus. This prevents the spread of infection. Then you get pain, which is called dolor in Latin. This is where you get stimulation of nerve endings. This is all part characteristic signs, classic signs and symptoms of inflammatory response. It's good to know that, by the way. So you get the injury, you get rubor, tumor, dolor, rubor and calor, and tumor and dolor. So you get redness, warmth, swelling, and pain. Okay, so this is what happens with inflammatory response. This is like you get punctured by a nail. So it goes through the first line of defense, and there's bacteria on the nail, and it penetrates a blood vessel. Now, these are, remember, there's white blood cells that are floating around all the time. And when they realize there's bacteria in the area, they send out chemicals to let everyone know that, let all the other immune response know that this is the area that they need assistance. Then you get this blood vessel will constrict to reduce loss of blood from this one. And then, so let's see, injury and immediate reactions, blood vessels narrow, vasoconstriction, the blood clots, and mast cells release chemokines and cytokines into the injured area. Then what happens, nearby blood vessels dilate. So other blood vessels that are intact, they dilate to let a lot more things in. So they let more fluid in, let more white blood cells in, increase vascular permeability, increase leakage of fluid, from exudate, forms the exudate. So they get a lot more platelets, they get clotting, and they try to wall off this area where it has invasion of bacteria. Then you have edema and pus formation. So you have this collection of fluid, the edema, swelling, infiltration by neutrophils, and formation of pus. This is where the troops start coming in. And then you develop this pus to try to bog down these microorganisms that are in this area so they don't get any further. Then you have resolution, scar formation. So you have macrophages, lymphocytes, fibroblasts, they migrate in, they come on in, they initiate the immune response and repair of injury. Then you have scar and loss of normal tissue. So it starts scarring up here, and your immune system is engulfing. You can see this dendrite engulfing this one. This one's coming after this one. And it's like, and this blood vessel is getting repaired. Then you talk about some of the chemical mediators of inflammatory response. I'm not going to ask you about these chemical mediators, but it just kind of goes through what we had just kind of talked about. And then diapedesis, migration of cells out of blood vessels into tissue. Chemotaxis is migration in response to specific chemicals at the site of injury or infection. So fever is an adjunct to inflammation. So fever is initiated by circulating pyrogens. Pyrogens actually reset hypothalamus to increase the body temperature. This signals muscles to increase heat production and vasoconstriction. And you have exogenous pyrogens. Exogenous means outside of. Exogenous, so it begins outside of our own body. So exogenous pyrogens are products of infectious agents. Like they secrete an endotoxin, and that endotoxin becomes a pyrogen, which will cause our hypothalamus to reset and increase our body temperature. Then we have endogenous pyrogens. These are pyrogens that our own body produces. And so these are liberated by monocytes, neutrophils, and macrophages during phagocytosis. So as they're eating up infectious agents, they're sending out signals to hypothalamus to increase the temperature. So here's some of the benefits of fever. Fever inhibits the multiplication of temperature-sensitive microorganisms. So some microorganisms, they work best at 98.7. So if you increase it, they may not work as well. It impedes nutrition of bacteria by reducing available iron. It also increases metabolism and stimulates the immune reactions and protective physiological processes. So with increased fever, things move faster. And so it's better for our body in fighting off infection if the temperature is a little bit higher. So let's talk about the second line of defense, the phagocytosis portion. So general activities of phagocytosis. First is to survey the tissue compartments and discover microbes, particulate matter, and dead or injured cells. Then to ingest and eliminate these materials, and to extract immunogenic information from foreign matter. So major categories of phagocytes. You have neutrophils, eosinophils, and macrophages. So neutrophils, they're general purpose phagocytes. They react early to bacteria and other foreign materials and also to damaged tissue. The eosinophils, they're attracted to sites of parasitic infections. And also they're attracted to antigen antibody reactions. Then you have macrophages. They're derived from monocytes. They scavenge and process foreign substances to prepare them for reactions with the B and T lymphocytes, which are part of the adaptive or specific immunity. So then you have development of monocytes and macrophages. So after leaving the bloodstream and entering into a tissue, a monocyte is then transformed by the inflammatory chemicals into macrophages or dendritic cells. Macrophages and dendritic cells can remain nomadic, which means they just float around on their own, or take up residence in a specific organ. This is where they're in the blood and they start off at monocytes. When they get into tissue, they can become a macrophage, which is a very big phagocytic white blood cell, or they can become a dendritic cell. So phagocytic recognition, how they kind of can recognize something that is not self. So you have protein receptors within cell membrane of macrophages called toll-like receptors. They detect foreign molecules and signal the macrophage to produce chemicals to stimulate an immune response. So this receptor is very specific to a particular microorganism. And so once this receptor comes across this foreign molecule, then it sends out signals to release cytokines, which will stimulate a further immune response. These are like the sentries. So this particular sentry runs across a foreign molecule, then it sends out the signal to say, hey, we have something bad around. We need you to mount a response. So you have chemotaxis, binding and ingestion. The phagocytes migrate to inflammation following a gradient of stimulant products. And then I'm not going to ask you about phagolysosome formation. So mechanism of phagocytosis, you have destruction and elimination. You have oxygen-dependent system, liberation of lactic acid, undigestible debris are released from the macrophage by exocytosis. I'm not going to ask you about that either. You have interferon. I'm not going to ask you about interferon. So, uh, what it talks about, well, I'm going to tell you in general in terms of if this cell gets infected by a virus, when the nucleus is compromised, the DNA in the nucleus is compromised, this virus takes over and starts assembling more viruses. At the same time, the nucleus starts sending out interferon. This interferon, as the cell is dying, is releasing interferon. This interferon is received by another cell, nearby cell, signals activation of the genes in this other cell, and if they're available, they send out antiviral proteins to help try to combat the attack that they know is coming, that has been attacked from the previous cell. That's like interferons send signals to try to warn other cells that there's viruses around that could attack them. I'm not going to ask you about complement, so we will bypass complement. But complement helps to kill the bacteria. I think that should be okay. So overview. Let's go through the overview. The host defenses. First, you have innate or non-specific first-line defense, physical barriers, chemical barriers, genetic barriers. Definitely know these different barriers and what could entail the different barriers. First line defense is a surface protection composed of anatomical physiological barriers that keep microbes from penetrating sterile body compartments. First line defense. Then you have the second line defense, inflammatory response, interferons, phagocytosis. So really we're talking about inflammatory response and phagocytosis. Second line defense is a cellular and chemical system that comes immediately into play if infectious agents make it past the surface defense. Examples include phagocytes that destroy foreign matter, inflammation which holds infection in check. Then you have acquired or specific defense. So that's the third line of defense. The B and T lymphocytes, then also the antibody cytotoxicity. So the third line of defense includes specific host defenses that must be developed uniquely for each microbe through the action of specialized white blood cells. This form of immunity is marked by its activity towards specific pathogens and development of memory.